we can't really have a discussion on the immune system until we have first considered the inflammatory response. Inflammation is one of the key responses that the immune system has to a particular possible problem. And just because there's inflammation doesn't mean there's actually a problem, but it means that there might be. So probably some sort of damage or some sort of injury has occurred that would trigger this. And I'm not going to go through this entire flowchart here in great detail, but what we do note is the release of inflammatory chemicals. And we'll talk about a couple of those here in a minute, but histamines are among the most significant of the inflammatory chemicals. They are then going to have a chain reaction, and they're going to cause arterioles to dilate, which means we'll have a greater blood supply to that particular area. Capillaries will become more permeable, meaning they'll leak more fluid out than they normally would. And that's also going to attract white blood cells to that particular area. There are several characteristics of inflammation that are called the cardinal signs of inflammation that are useful, and you can sort of diagnose that there is inflammation going on when you see these characteristics. And that is what you see here in the red: heat, redness, pain, and swelling. The heat is going to come from increased blood flow to the area and therefore increased metabolism. And the thought of the body at that point is, if you can increase the temperature here, it's sort of like a localized fever. You might be able to cook the pathogen by increasing your metabolism. The redness is a natural result of increased blood flow to the area, and in this case blood's flowing there, but it's not flowing out as fast as it's flowing in. Because of that swelling that's occurring, there's going to be some pain, perhaps, and the pain is because nerves are being compressed. So when you have heat, redness, pain, and swelling, those are a surefire indication of inflammation going on. And there's a natural inclination to try to turn off inflammation by taking an anti-inflammatory like a Benadryl or something like that. But as long as it's not impairing function, then it really should be left alone, because the whole point of the inflammation is to keep whatever problems there might be a localized problem and not allow it to become a systemic problem. And I know you've heard me say this multiple times throughout the different topics, but it's so incredibly important. Keep local things local, and they don't cause problems other places. Those inflammatory chemicals we mentioned, again, histamine being the most significant, but there's a couple of others here as well that you can take a look at. And they're all going to involve some piece of the inflammatory response. One of the things that's going to happen is the mobilization of phagocytes. And phagocytes are cells that eat foreign particles, sort of like janitors. They're eating anything they don't recognize, they're cleaning it up, so any possible infection that's there is possible to be eaten by a phagocyte. So what's going to happen in that process is the inflammatory chemicals we already mentioned would attract white blood cells. They're then going to go through a three-step process, and one of those is leukocytosis. So entrance of new white blood cells into the blood that were being held in the bone marrow. So you increase your white blood cell count. Step 2 is called margination, and what happens there is the capillaries nearest the site of inflammation will allow the cells to stick. So the white blood cells are going to stick to the walls of the capillaries in that region, and then they are going to diapedese. That is, they're going to squeeze through the capillaries, and if you remember the capillaries, they would have some of those intercellular clefts and some other large openings there that these guys might be able to squeeze through, and then they're going to go out into the surrounding tissue where that problem might be, and hopefully be able to clean up whatever kind of infection, or potential infection we should say, might happen. So that leads us to step 4, the idea of chemotaxis. And that is they're going to follow the concentration of the inflammatory chemicals, and they're going to move towards higher concentrations. So the closer they get to the area of problem, the higher the concentration of inflammatory chemicals should be, resulting in a way, a dog following a scent trail. In this case, white blood cells following the smell until they find the area where the potential infection is most likely located. The world of viruses is especially complicated for modern health because we don't have a great deal of things that we can do about a viral problem. We can try to develop vaccines, and that's a wonderful thing, but we just can't develop a vaccine for everything. So one thing that we can do to try to treat after the fact, so the vaccine was a preventative, not an after-the-fact treatment, is we can perhaps use this idea of interferons. So if we would follow the process through here, in step 1, the virus enters the cell. And when the DNA of the cell mixes with the DNA of the virus, that turns on interferon genes, so genes that produce this protein interferon. That is then released from the cell and then attaches to a interferon receptor on another cell, which then is triggered to make antiviral proteins. So cells already have the ability to make proteins that will interfere with viral reproduction, with viruses entering a cell, basically just slow up the process. But they aren't really stimulated to turn that on to a great degree until interferons are present. So really a couple of cells have to suffer for sure before the antiviral machinery gets turned on. This is a naturally occurring process, but in healthcare what we often do is we provide a massive dose of interferons that would, to a great deal, stimulate the body's natural antiviral processes. So typically we say if you get a viral problem, you have to wait until your body figures out how to defeat it and hope it does. And if it doesn't, it will kill you. So in this case, the addition of more interferon will jumpstart, will supercharge your body's natural processes, trying to help it work better than it would perhaps on its own. This is also a possible application for some cancers. And so in some cases, interferon seems to be able to activate anti-tumor DNA and production of proteins as well. And so this is a treatment idea that's been around for probably 25 years or so, but it still, I suspect, hasn't been developed to the greatest of its potential yet. Complement is another complicated system that involves dealing with problems as well. And so there's a couple of different ways that the complement system can work. We can have the classical pathway, in which the cell that is potentially the problem is coated with antibodies. And that then identifies that cell as being a problem. It also allows phagocytes, so those cells that would eat problem things, sort of a guide or a map to find them because they're what we would call opsonized. And basically that means that they're coated in these antibodies, and that encourages the cells that are eating foreign particles to have a bigger appetite and to specifically focus on the cells that are coated in that particular protein. The lectin pathway is going to be sugars attaching to that problem thing's surface and further enhancing their consumption by phagocytes. What often happens as part of this process as well is it enhances inflammation. So the activation of the complement system increases the inflammatory response. So it basically is doing that to try to get more white blood cells out there more quickly as it's labeling these problem things and hoping as soon as they're labeled, then they can immediately be consumed. One other possible functionality of the complement system is the formation of a MAC, a membrane attack complex. And basically what happens here is a number of these complement proteins will actually puncture the target cell's cell membrane, forming a hole. So basically it's drilling a hole through the cell membrane of this bacteria or this problem thing that's identified, and then basically it bleeds to death because it's been stabbed with all these holes and it can't survive that experience. So the complement system is enhancing the other white blood cell functionalities going on, enhancing inflammation, and poking holes in target cells to kill them that way. Those things so far have been part of the innate system, or the automatic immune system that's there automatically. It's not overly specific, so it doesn't have to learn new things. It's pretty much multi-purpose, and any of that kind of problem that comes along, it does its best to deal with. In the world of adaptive defenses, this is where the immune system needs to learn things. And so it's going to hopefully learn a particular solution to a very particular problem. This is where we get the idea of immunity, and this is where vaccines come into play, is to help you get that learning without actually having to deal with the specific problem itself. So what we see here is an antigen. And an antigen is anything that triggers the body's adaptive immune system. So it's a potential problem that's been identified by the body and it's triggered the adaptive response system. Really, an antigen usually becomes something that is not part of the human body. So anything that has not been labeled and identified as belonging to that person can then be labeled as an antigen. There are self-antigens, and there are what we might call foreign antigens. And self-antigens is basically a protein structure that is not foreign to you, so it specifically belongs to you, but it belongs to you, not the human sitting next to you. So if you got, let's say, a heart transplant, that came from another human, and while it is a human heart, your body would look at it and say, I don't recognize the MHC proteins, the major histocompatibility complex proteins. I don't recognize those. So that might be a heart, but it's not my heart. And then your immune system would try to kill the new heart. Obviously, that would be problematic. So in this case, the antigen process is labeled as human, but it has to be a particular, specific human in those cases for things to be compatible. This makes tissue and organ transplants complicated because very often it's impossible to find someone who's very, very, very close to being exactly like you in terms of self-antigens. And unless you have an identical twin, it's probably not going to happen. But they will do their best to find as close of a match as possible, meaning your body's self-antigen process is not going to be as dramatic as it would be if it were someone who was very much different from you. So when they're looking for a match for someone for organ or tissue transplant, they're looking at these self-antigen processes, these major histocompatibility complex proteins, and trying to find someone who's as close as possible so your immune system has minimal response to that. You will still likely need to be on anti-rejection medication for the rest of your life. And basically the point of that medication is to turn off your immune system response so that it doesn't destroy this new piece that you got. Now, there is a problem with that because if you've turned off your immune system response that much, that means you're now susceptible to other things that come along and might become a problem. So there's a trade-off there. I suppose the alternative of dying without the new piece is probably not desirable, so we'll go with suppressed immune system as maybe the better option there. The cells involved in the adaptive immune response include B lymphocytes, or B cells, T lymphocytes, or T cells, and antigen-presenting cells. So let's take those one at a time. Let's look at the B and T lymphocytes first. And they must acquire two functionalities before they're effective. One is they must achieve immunocompetence. And what that means is they must learn how to identify or to recognize a specific problem. And at that point, they're specialized. And they would only recognize that problem, and they wouldn't recognize any other particular antigen that might come along because they have achieved immunocompetence. They're up to speed on one particular problem. If you had 12 different possible problems in front of you, you would need 12 different immunocompetent lymphocytes to be able to handle that. The second thing that these lymphocytes must achieve is self-tolerance. They must be able to recognize what belongs, so recognize the cells that belong here, and leave it alone. If they don't achieve self-tolerance, then your lymphocytes basically attack your own body. And for those of you who have allergies, that's exactly what happens, is your self-tolerance isn't what it needs to be. And in the process of responding to a particular problem, the body over-responds and starts attacking you as well. So in other words, it fails to identify and tolerate your own body in that immune response. And you would know then what those symptoms of the allergies are and how incredibly inconvenient that can be. There are a couple of different steps that they go through to achieve this. So in this particular scenario, T cells would go to the thymus. And we'll see the thymus a little bit later on as endocrine system function. But right now we're talking about it teaching white blood cells what to do and how to identify things. So the first step is called positive selection. And in positive selection, the T cell must recognize the major histocompatibility complex proteins. So in other words, it must self-recognize this belongs to me, and then it can go on to step 2. But if it fails to recognize the MHC complex, then that particular cell is going to die. If it passes the first test of positive selection, then it moves on to the second test of negative selection. In this case, the T cell must not respond to a self-antigen. So it needs to recognize this particular piece is me. It's specifically me. And if it is capable of doing that, then it goes on to be a successful, long-lasting, hopefully, T cell. But if it fails to ignore self-antigens, then it will die at that point. So it's a 2-step test. If you survive the positive, you go to the negative selection. If you survive that, then you become a functional T cell. Otherwise, you die and the process stops at that point. If we look at the B and T lymphocytes here, we can see that there are some differences between them in terms of what they do. So let's take a quick look at that. B lymphocytes are involved in humoral immune response. So that means in the fluid, in between cells and tissues. So floating around loosely in the body is what's going to be dealt with by B lymphocytes. They're also going to secrete antibodies, and we'll talk about antibodies a little bit later here. Their primary targets, bacteria, fungi, and some parasites. T lymphocytes are involved in cellular immunity. So this is going to deal with problems within a particular cell, not necessarily out there floating around in the fluid. They are not going to secrete antibodies, and their primary functionality is going to be on virally infected cells and cancer cells. So within a human cell problem versus outside of a human cell problem with the B cells. There are going to be a couple of end results here. B lymphocytes become plasma cells, and T lymphocytes can become cytotoxic T cells, helper T cells, or regulatory T cells. And so there's a little bit of different specialization and functionality that's going to go on there in that process. Both types, however, will form memory cells. And that means that once you form this particular kind of cell for this particular kind of problem, the memory cell should keep that response recorded. And if that particular problem presents itself again at a later point, it immediately pulls out the solution and says, here's how to fix that. Make a bunch of it, and the problem is immediately solved. Meaning most of the time when you have problems like this, that the body will remember that problem, and you'll probably never experience that same exact problem again. So let's say you get a particular thing, and let's just say, since everybody's thinking about the coronavirus right now, Let's just talk about that one. So as your body would deal with this virus, mostly with T lymphocytes, but some B cells might get involved as well, they would make memory cells. And that would basically say, here's how we killed this particular virus. If you were then exposed to coronavirus again at a later date, your body would immediately pull out that memory, make that particular cell that was most effective at killing the virus, and immediately solve the problem. And you would never probably ever have coronavirus a second time. It's a little bit early in the process to tell for sure exactly how long this memory will last and how long you would then have immunity to that particular problem. But many viruses are a one exposure and then you're good for the rest of your life. Chickenpox, for example, it comes from a virus that many of you probably received a vaccine for. And I think that's really not fair. I think you should have all had chickenpox and suffered greatly from it. I didn't get chickenpox until I was 18. And I'm of the generation that didn't have a chickenpox vaccine available. My body then just had to figure out how to deal with that. It then developed that memory, and I will probably never have chickenpox again. Those of you who got the chickenpox vaccine developed the memory by dealing with the vaccine itself, which is a kind, gentle way to learn and develop a memory. And then from that point on, you probably will never get chickenpox either. So if we look at the B cell process here, I'm not going to go into great detail here, but the primary response and a secondary response is what we want to get into here. So we see with the primary response, the antigen, this problem thing, is identified. It's a specific B cell deals with it, and then those particular B cells proliferate. They divide, they clone themselves, making lots and lots of themselves. Most of those become plasma cells, and the plasma cells are going to secrete antibodies. Antibodies are going to help tie up any of those antigens that they find floating around the system. But a couple of those cells will become memory B cells. And those go into the memory banks, and that's what's going to remember, perhaps years later, if you're exposed to, let's say, this chickenpox thing again. And next time around, look what happens with the plasma cells. It took a little bit of time to form plasma cells in the primary response. And we didn't form that many. But in the secondary response, when we pull out that memory and activate it and use it to produce the proper solution, look how many more plasma cells get produced. Way more plasma cells are produced in the secondary response that can be months to years later, and far more antibodies are secreted. So a much more powerful and much longer lasting response occurs with a secondary response. Let's look at that a little bit differently. Let's say that this one's probably a vaccine because we're looking at pretty close exposure to each other. But let's say the first exposure to a particular antigen, a particular problem, is on day 0. And by about day 5, you start to see antibodies being produced that are dealing with that particular problem. And it lasts perhaps for a couple of days, maybe a couple of weeks, and then those levels of antibodies drop down to practically 0. So we didn't get a huge response, and it didn't last that long. But a secondary exposure to that same antigen, in this particular case about 20 days or so after our primary response was at its peak, look at what happens to the response there. We get a very rapid increase of antibody production. It is far more massive in terms of numbers of antibodies produced than we saw in the primary exposure, and it lasts for a much longer period of time. In this particular case, it looks like it would probably trail off, but even when we get out to the number of years later, we would still have some degree of antibody presence there. And so that would ensure that we developed good memory. And what we found with a number of vaccines is that one exposure gives you what we see in the primary response. But a secondary exposure, so that would be called a booster shot in the world of vaccines, gives you this secondary response, so you get a bigger response, a longer lasting response, and more memory cells get made. So really you're repeating, you're rehearsing you're studying it over again, and the more often you study something, the more clearly you remember it. So in this case, a couple of exposures to something, and then at that point we think that you're probably covered for the rest of your life on that particular thing. In the world of humoral immunity, so that was in the fluids. That was primarily our B cells, remember. There's going to be two different kinds of humoral immunity, active and passive. Active humoral immunity is maintained, so it's going to be something that keeps around, it stays around, it keeps going, it keeps replenishing itself, hopefully, and that can be acquired one of two different ways. Naturally acquired would be you get the pathogen, you get sick from it, you deal with it, and you acquire the immunity and the memory. And again, hopefully that's maintained for your entire life. The second option is the artificially acquired, and that's where the vaccines come into play. So a number of you probably get a flu shot every year. The idea of that flu shot is to give you artificially acquired humoral immunity to those particular mixes of flu viruses. Now, a vaccine is going to be a dead or an attenuated pathogen. So a dead virus is basically an inactivated virus. And saying a virus is live or dead is sort of misleading because viruses are not living things. They're just a bundle of genetic material wrapped in protein. So this really leads to the problem of how do you kill something that's not alive? In this case, we just have to develop the ability to recognize it and to physically destroy it. So the dead one is a completely inactivated version, probably pieces, not necessarily a full virus. Sometimes pieces are good enough for your body to learn from. An attenuated pathogen is going to be a weakened one. So it's been punched a few times, but it is functional. It's just weak enough that it most likely is not going to give you the problem, but still give you, again, the memory and the protection. Passive humoral immunity is only present while that particular product is in place. So naturally acquired humoral immunity would be things like the antibodies that would pass from mama to baby in the placenta, and we'll talk about the specific antibodies later on here, or transmitted to the baby in, let's say, breast milk. So it's important to breastfeed the baby if at all possible because that's giving mama's immune system, or at least mama's immunity, mama's antibodies to that particular problem being transmitted to the baby when the baby probably can't make its own yet, or certainly is not going to be good at making its own. Artificially acquired passive humoral immunity is when antibodies are injected. And this might occur if you're going to go, let's say you're planning to go to Africa, and you know that malaria is a problem in Africa, and you don't want to get malaria. And what you can do is you can go to the doctor and they can give you an injection of antibodies against malaria, and that will give you a couple of months of protection. But once those antibodies break down, whether it was artificially or naturally acquired, once those antibodies break down and you're not getting more of them, then you no longer have that immunity. So passive immunity is temporary. It's only there for as long as those antibodies that you got were functional. But in the passive scenario, your body's not actively making more. It just took some for free, and when they break down, that's all there is, folks, and then you're no longer protected. So if we look at those antibodies here, or immunoglobulins, immunoglobulin and antibody is the same thing, but for some reason somebody decided that it needed to be called something else as well, I suppose just to make things more complicated. So there are 5 different categories or classes of antibodies. The first one is IgM. IgM, or immunoglobulin M, is part of a primary response. So if you look at someone's blood and there are high concentrations of IgM, that tells you you're probably in the middle of or shortly at the end of a primary immune response. It's readily involved in the complement system. IgA is found in secretions of the body, saliva, sweat, breast milk, things like that. So IgA is one of those that would be transmitted from the mama to the baby in the breast milk. And here's what it does that's so wonderful for the baby, is that IgA prevents pathogens from attaching to surfaces. So the more IgA you have, the less likely it is a pathogen can stick, whether it's to the skin, whether it's to the mucous membranes of the mouth, the nose, the esophagus, that sort of thing, or the walls of the intestines. So what that means is if a baby does ingest a particular pathogen and there's a lot of IgA present, it most likely will flush through the system without being able to attach and get its way into the body and into circulation. So this prevents things from entering the body, and that's really helpful, especially again for that baby that doesn't have any functionality on its own. IgD is an antigen receptor on B cells. IgG is the most common antibody that you would find in the plasma, accounting for 3/4 or more of all of the antibodies in circulation. If you find a lot of IgG present, like we found in IgM, that suggested a primary response. IgG suggests either a secondary response or it's very late in the primary response. It's also going to be involved in the complement system as well, but more of a wrapping things up or doing things much more aggressively the second time around. And it's going to be a protector from a wide variety of things, from bacteria, viruses, toxins, the list goes on and on. This one crosses the placenta. So the IgA that was crossing via breast milk was not crossing the placenta, but IgG was crossing the placenta. So you don't have everything crossing the placenta. You don't have everything going in breast milk. But again, any level of protection you can get is certainly worth it. IgE is going to be involved in the inflammatory response, so releasing histamine and other inflammatory chemicals. The allergic reaction process is going to be involved here, and lots of different places you could find this being released. If you find high levels of these, that probably means there's been some severe allergic reactions going on, or perhaps, and this is quite comforting to you, I'm sure, if you don't have a lot of allergy problems going on, but you still have high levels of IgE, you probably have some sort of chronic parasitic infection in your digestive system. I'm sure that's a delightful topic to consider. This adaptive process includes a number of different functionalities here. So the antigen comes along, the antibody comes along, one of these 5 Igs we've been talking about here comes along and attaches to that antigen. Now the antigen is identified, and it does a couple of different possible things. It could either fix it and activate the complement system, so in other words, stop it, and then call for the complement system to come along and poke holes in the membrane, which would then lead to the cell lysing or dying. It would also enhance the inflammatory response. So it would cause more inflammation to come along, and that would then bring more white blood cells and further seal off the area. If we went the other direction with it, we would inactivate that particular antigen by neutralizing, agglutinating, or precipitating. In neutralizing, what it's going to do is wrap that particular virus or bacterial toxin in antibodies, which basically covers up their functional area. So they're still there, but they can't actually cause problems because their actual harmful components are covered up with antibodies, and they'll eventually then flush out of the system. So they're neutralized. Still present, but no longer harmful. Agglutination will cause antigens to stick together and form big balls. So this is sort of like coagulating or collecting them up in a ball rather than having them floating freely around the place. So this kind of helps to isolate problems and make them one particular area all bound up rather than, again, floating all over the place. If you have wrong blood types that mix, this is the kind of reaction that's going to occur. So when we talked about blood types and we talked about anti-A and anti-B, we would have talked about that, and then the agglutination would be if the anti-A caused the A to clump together, or the anti-B caused the B to clump together. That would be an agglutination reaction, and that's an immune system reaction. The third option here is precipitation. So what happens here is antigens that are soluble, they're dissolved, they're readily moving in the fluid, will be bound up by the antibodies, completely surrounded, and removed from the solution. So they're no longer in the solution, they're sort of like agglutination in a way of gelling it up and taking it out of solution. So sort of like removing salt from water is removing it from that soluble condition, and then precipitating or sort of settling out and then filtering, probably being taken out by the lymph nodes when they pick up that fluid. The fixing and activating of the complement is not only is going to enhance inflammation, it's going to enhance phagocytosis. The inactivation with antibodies, whether it's neutralization, agglutination, or precipitation, is also going to enhance phagocytosis. So anytime you have those antibodies doing something, it's going to encourage phagocytes to have a bigger appetite and to more aggressively eat anything that they find the antibody attached to. So in a way, the antibody becomes somewhat like a sign that says, I'm a problem. Come get me. Eat me first, please. From the T cell perspective, it's going to function in a somewhat similar way to the B cells that we've been talking about thus far. But again, we're looking here at a direct cell problem, not something necessarily floating around in the fluid. So what we're going to have here is a couple of different kinds of T cells get developed in this process. And we'll talk about CD4 and CD8 cells. CD4 cells are going to be cells in this developmental process here that are helper T cells. Sometimes we'll call them a helper T cell. And that's one thing that they can produce. And those helper T cells are going to activate B cells, so make the B cells out in the fluids more active. They're going to activate other T cells, and they're going to activate macrophages. And macrophages are cells that are sort of a phagocyte, but they have an incredible appetite and they have an incredible aggression that they pursue foreign particles with. So these helper T cells basically stimulate the rest of the adaptive system to be more active and functional. So if you have helper T cell levels increasing, everything else in the adaptive system is going to get more aggressive very shortly after that. So sort of like it turns on everything else. CD8 cells become cytotoxic T cells. And those are going to sometimes be called killer T cells because they will kill any cell that is labeled as containing a foreign particle. So if you watch the video, the other video here in the unit material, it's on viruses and vaccines. And they talk about killer T cells and helper T cells there. And so that's a pretty good description of how they're going to work. But Any cell that has a foreign particle in it probably sends a protein out to the surface of the cell that's basically like a name tag or a flag that says there's something foreign, there's something weird going on in here. And if a cytotoxic T cell sees a cell with this label as containing a foreign particle, it will attack it and kill it. So that's how it deals with problems inside a cell, as it reads that flag saying there's a foreign particle here, and then it kills it. There are a number of different chemicals involved in stimulating these processes, and we've mentioned interleukins, or rather we've mentioned interferons, and that's going to be activating some of these other functionalities here and helping turn on cells with their normal functionalities. Interleukins are also going to be involved in stimulating these different processes here. And so if you just look through this list, there's a long list of different interleukins there, and they're all going to stimulate something. They're going to enhance something. So really what we're saying here is that, and if you look on down here, we also have suppressor factors and tumor necrosis factors. These are all stimulating your body's natural processes. So your body naturally looks for tumors, and if it finds it, kills it. Your body naturally looks for viral problems. If it finds it, kills it. Your body naturally looks for bacterial problems. If it finds it, it kills it. But all of these factors here are going to stimulate. They're going to encourage. They're going to enhance those natural functionalities. So a lot of modern healthcare nowadays is not necessarily giving you a medicine to kill a problem, but it's giving you a medicine that will do what we're seeing here. It's going to enhance, it's going to stimulate, it's going to magnify your body's natural responses. So in other words, it's just helping your body do what it already does, but do it better and do it more aggressively. And what we're finding with that sort of process is your body was already pretty good. If we give it additional enhancement and stimulation in providing these kinds of treatments, then your body, in many, many cases, is able to deal with it on its own without introducing so many incredibly toxic pharmaceuticals. Anytime you can help your body do its own job rather than introducing a harmful compound to the system, you're better off. Here's an example of a cancer cell, and it's been identified as a cancer cell, and this cytotoxic T cell is killing it. So what it does is it goes down and it binds to that particular cell, and it releases these enzyme proteins called perforins. And what those perforins do is they act just like the complement system. They poke holes in it, and it basically lyses and bleeds to death at that point. They can also introduce some other enzymes that are very strong digestive enzymes inside. So once you've poked the holes, then you can dump these other digestive enzymes inside, which destroys it from the inside out. It's a nice little image there of that cytotoxic T cell getting ready to kill that cancer cell. So many of us, probably all of us, have had cancer multiple times in our life, but thus far, for many of us, our bodies have been able to label it as a problem and send out an appropriate cytotoxic T cell to kill it. So most cancers, I would propose, are self-cured by the body before we even realized that it was there. Here's sort of a summary, if you will, of the adaptive immune response. The B cells, the T cells, and the different kinds there. There's also a group of cells called antigen-presenting cells. Antigen-presenting cells are going to grab hold of a particular problem. They're going to eat it, and then they're going to take a piece of it and wave that up in the air and say, see this piece? This piece came from a foreign particle. It's a real problem. If you see anything that has this piece on it, kill it. And so basically what it then does is it's told the T cells, here's the problem we found, here's what it looks like, go out and find the rest of it. And so when the antigen-presenting cells work like they should, they greatly stimulate the immune response, showing T cells, here's the identified problem, go out there and look for more of this. Most of the other things here in this chart we have talked about already, but take another look at this. It's, again, a good summary of all the pieces and parts, and at least to some degree, what they're doing. You're going to read about allergies in your reading assignment, but I just wanted to throw this out there as what's going on when you have an allergy reaction. So the antigen, that foreign particle, is detected. And for many allergies, it's detected in the respiratory system because that's the part of our body that's most open to the environment. And then your body recognizes the antigen, produces plasma cells, produces antibodies, which then attach and stimulate the immune system response. So often, the first time you're exposed to something that you have an allergy to, it's a relatively mild response or problem because just like with the primary response of your immune system for everything else, it's not that strong and powerful. But in the world of an allergy, that's now labeled as you are sensitized. So the sensitization stage was the primary immune response, the first time the body saw that particular thing and, in this case, overreacted to it. Secondary responses, just like with the normal immune responses, are going to be much more dramatic and much more aggressive. So this is where the idea comes in that the more often you're exposed to the allergy, to some degree, the worse the allergy gets. Because again, the immune system is learning. And in actuality, it's getting more aggressive and better at dealing with the problem. But again, really our issue was it failed to properly recognize self. So the self-identification process was faulty. The self-tolerance wasn't working, and so it then attacks other problems. So in the respiratory system, it's going to cause constriction of bronchioles. That's the small tubes leading to alveoli. So if those are constricted, you're going to have a hard time breathing. If mucus release is enhanced, and again, that would be okay normally because that's going to help capture pathogens, help capture antigens and flush them out of the system. But if we already have constricted airways and we increase the release of mucus, that further constricts and makes it more difficult for the airway to remain open and for you to breathe. And again, the capillaries become more leaky, so you have accumulation of fluid in the respiratory system. So the problems that you're experiencing, you might also have watery eyes, runny nose. That's because of increasing amounts of fluid in the system as well. Coughing is because of respiratory spasms in the bronchioles and the plugging up of mucus of the system. And so that respiratory distress that often comes along with an allergy response, the immune system overreacting, causes that because of this sequence of events. And normally that sequence of events would be okay, but in this case it just does it too much. Some other things you might find hives, you might find rashes and things like that. And so you can read more about those in the reading assignment for that. But this is what causes those responses, is the plasma cells releasing antibodies and all this triggering histamines, and the regular inflammatory response. But in this case, again, failing to leave your own cells alone and only attack the problem, only attack the antigen. And as long as you only attack the antigen, again, not a problem. So some people have allergies that it seems like they're allergic to everything, and other people seem to have no allergies whatsoever. And I'm more in the category of no allergies. A couple of years ago, I started noticing, though, that if I'm outside around a lot of fresh-cut grass and things like that, I might have a little bit of runny nose, but it's nothing major. It's a very mild reaction. Nothing along the lines of anaphylactic shock, for sure. My wife, on the other hand, is allergic to a lot of things and has a lot more significant responses to it. So her body is sensitized, it's overly reactive, and mine, in a way, is a little bit more of a laid-back system, I guess. So people who have allergies have very reactive immune systems, but I think we can say that being reactive is not always necessarily a good thing. If you're overreactive, it's a problem. But we also don't want to have a system that's too laid back and ignores problems that are there. So there's a happy medium there. And probably most people have a system that's mostly in that happy medium. But more and more we're seeing more people with allergies. And whether that's because we're becoming sensitized due to our diet and other environmental pollutants and exposures, or maybe it's that we've always had these problems and they're more accurately being labeled now as being allergies. I don't know, but either way, it does seem that allergies are increasing in the population. So, again, whatever that means, I don't know, but certainly it means more inconvenience for everyone involved in the process. Hopefully you learned a lot. Hopefully it all made sense and was clear. Have a wonderful day, and we'll see you next time.